Okay, so welcome back to the second class of E443. As always, I need to remind you that the course is being recorded and live streamed. That is going to be made available. So that you're aware of that. I'd like to start out by, let's see if my cursor will work. Reminding you just a bit about the theme of this course. The theme of this course is multicellular organization. And we're going to be focusing on simulations in which you have substantial numbers of cells. That might be anywhere from a few to tens of thousands of cells or hundreds of thousands of cells. And we'd like to know how those cells interact with each other and behave to build tissues in the first place, to maintain them, and to fail to maintain them in illness. And potentially also how we could use that information to build and design tissues with controlled properties, tissue engineering or biomedicine. One area that definitely could be of interest would be organ-on-a-chip systems. So Feng Guo in our department builds organ-on-a-chips, organoid cultures. And at least in principle, one could build simulations that effectively are at that scale, where you simulate all of the cells in the chip. We'd have to talk to him about which particular ones he's simulating, but I mean, he needs simulating, but it's certainly possible. So those are some of the things that we thought about. Basically, the simulations that we're going to do have these three main length scales associated with them. The networks, the molecular networks that control cell behaviors, the cells and the things that the cells do to each other and their environment, and then the tissue scale organization of chemical fields, forces, boundary conditions, and so on that define the extracellular environment and how tissues organize. In some cases, we'll put that in the context of a whole body model, typically also a very simple non-spatial one, although one could take, for example, last week we had this very nice talk by somebody from Alan Perlson's group where they were doing models of COVID infection, and the experimental data, the clinical data that they were working with had data for COVID levels in the upper respiratory system and also in the lungs. And so one could imagine building a patch model like the one we showed last week, where you have a patch model of the upper respiratory system, a patch model of the lung, and connect them, and taking that ODE model, non-spatial model, to a compartment model that they built, and turning it into a spatialized model with two domains. So it's possible to build higher level organizations as well. But typically the volumes of tissue we're talking about would be on the order of a centimeter by, centimeter, I wish we could do, a millimeter by a millimeter. A little bigger if it's quasi 2D, a millimeter cubed. Any questions about that so far? And then we had lots of movies, because the output of these simulations are movies. And here I've just aggregated. For some reason, a lot of these movies didn't play last week. I still don't understand what happened. So I'm showing you some of these movies. One of the things that points to this montage was to show the variety of problems one could work on with this kind of modeling, and also the different levels of spatial resolution that you could have. So at the top here, we had a single cell crawling, and that cell was represented in a lot of spatial detail. On the other hand, this somatic cancer evolution, the individual cells have relatively little detail. this drug dosing in liver model here on the top right, the individual cells actually aren't represented at all. You're representing volumes of the liver tissue. And each simulated, quote, cell represents many real cells. And so again, depending on the kind of problem you're working on, you'll pick different levels of spatial resolution, different numbers of cells, different kinds of phenomena that you want to represent. The single cell crawling model is a true three-dimensional model. This angiogenesis model is a true three-dimensional model. The cyst model and the tumor angiogenesis model are pure 3D models, true 3D models. This liver model here is 2D only. It's a single, it's 1 voxel thick. That's true also for the somatic cancer evolution model. This viral infection model is what we call 2.5D. It is two-dimensional layers that sit on top of each other with a particular spatial relation. That gives you a lot of the benefits of a full 3D model and runs a lot faster. And so that's sort of a compromise between full two-dimensional, three-dimensional modeling and pure 2D model. So today, I'd like to start out by just going around the room and having people introduce themselves. Again, I realize a lot of you know each other already, but there's some new faces, or at least new Zoom links. I'd like you to just briefly introduce yourselves. And if you have thought at all about possible project areas or topics, can be very, or just areas of interest for you, that would be great. Why don't we take a few minutes to do that? And then we'll jump in and start working with CompuCell. I'll tell you a little bit about how CompuCell represents the things that we've just seen, because some of the ways that CompuCell maps real-world concepts to mathematics and then to computation may not be so familiar to people. Once you get the hang of it, it works pretty easily, but some of the ways it does things are not perhaps so obvious, especially if you're not coming from a strong, a lot of physics background. If you've had a lot of physics courses, the things CompuCell does will feel fairly natural. If not, you may take a little bit of playing to get used to them. Most of the time, the details won't matter that much, and the math won't matter that much. When it does, we'll talk about it. So I was going to turn it over to you and ask you to go around the room and introduce yourselves briefly. If you've thought about possible projects or topic areas of interest, please say a few words about it. If you want to screen share, that's fine, but you certainly don't have to. If you need the screen, let me know and I'll release it to you. Otherwise, just a few words. Why don't we start? Tyler, you want to take it away?
Sure. My name is Tyler Hanna. I'm a junior in ISE and concentration in bioengineering, and also a minor in math. And regarding my project for this semester, I've looked at the CD8 T cells, the recruitment and maturation of those in lymph nodes. And since I did a project over influenza and the immune response last semester, I think I want to focus more on the immune system and kind of go into that kind of area.
That's great. I mean, there are a number of possibilities. One could actually try to model a lymph node. There's some beautiful work by Denise Kirschner using agent-based models, different ones, to try to model lymph nodes. When we taught our CompuCell course last summer, there were a couple of people who were interested in modeling the thymus. So the thymus is responsible for sort of early stages of adaptive immunity, and has some interesting behaviors, although it's not really that active during adult adaptive immunity. It generates the repertory of T cells early in life, and then shrinks, becomes less active in adults. One of the reasons that adults have problems, especially very old people have problems with immunity, is because they don't have an active thymus. And of course, TJ's worked a lot on more detailed models of immune response, and so he might be available to talk to you about those kind of problems. So I think that's a great problem. There's also the possibility of doing some of the things we did last semester, which is TJ's and Josh have both worked on this idea of taking models that have been developed in non-spatial contexts, like this one of Alan's, and then turning it into validated spatial models. I think those are things actually one could develop and publish. Whether you could get the whole paper done in 3 months, I don't know, but within a few months. Devin.
Yeah. Hi, guys. My name's Devin Keller. Pretty much in the same boat as Tyler. I'm a junior in ISC, specializing in bioengineering and a minor in math. I haven't thought a lot about my project yet. The idea of cancer is something that's interesting to me. And then I also liked working with viral dynamics last semester in your class. So maybe something within hosts with viruses sounds interesting also. Thank you.
Drew?
Hi, I'm Drew, also a junior in ISC in bioengineering. For my project, I'm still kind of stuck between a few things, but I was, I don't know, interested in looking at the 3D representation of like, you know, a differential equation sort of simulation, and I don't know, I want to look more into that. Either that or like something related to drug delivery.
So, okay, so when you say a three-dimensional representation of a differential equation, are you thinking about, do you have something, an example, any sort of example in mind? That could mean a lot of different things. It's a big topic, and so.
I mean, yeah, well, I don't know, because like the COVID simulation thing that we ran, like that was, we just saw a bunch of graphs. I don't think it had any three-dimensional components to it or anything, but I want to visualize something and compare the two.
Okay, well, there's certainly a variety of differential. I mean, there are a lot of everything we're doing are numerical approximations of differential equations of one sort or another. Well, maybe not everything, but 90% of what we do. And in particular, we have diffusion, which is critical. So how molecules move in the environment. Those molecules could be very large, like viruses, and so the solution to the diffusion equation is a core part of these kinds of solvers. And then the control, the molecular level control, is represented usually with ordinary differential equations. It could also be stochastic. And so definitely there are interesting questions about how spatial organization occurs. There's some classic models of things like bacterial organization in 2 and 3D to form structures. There's a famous example of Dictyostelium discoideum, which is a multicellular, well, an organism that alternates between unicellular and multicellular organization. And there's some beautiful papers from 20 plus years ago using the same methodologies that we use that we've never really replicated. It could be interesting to do. So there are a variety of interesting possibilities there. So keep us posted on how your thinking develops. That'd be great. Jeffrey.
Hi, I'm Jeffrey. I studied math and physics as an undergraduate, and now I'm doing a master's in the ISC department. I'm kind of interested in neuroscience now and would like to continue with that. But also I am interested in fluid dynamics, and I think it was Andy who was mentioning fluid dynamics in his thing. So I haven't really decided yet, but I'm taking a class with John Beggs in the physics department right now. So I'm going to try and see if I can use CompuCell for our project in that class, and then try to do something with fluid dynamics with Andy over here, just so I can kind of get the best of both of them. But I haven't decided at this point yet.
As I say, you could certainly put neural oscillator simulations inside of cells, and then couple them and see what happens in a spatial context. I mean, the one thing that you don't get a lot of usually with these neural systems is the cells don't move around a lot. Typically, once your neurons are fully differentiated, that's more or less it. They don't crawl around and reorganize. During early neuronal development, there's a lot of dynamic movement, although the complexity of that is really hard to model. There's some beautiful papers by George Henschel going back almost 30 years on neuronal outgrowth and organization. I don't know how easy it would be to put those into a CompuCell context, but it's possible. But certainly neural synchronization, the kind of things that you talked about before, that's something one could do in a spatial context and study the effects of that. Fluid flow, Andy's code is good at that. Our code does it. It's not terribly elegant, but you can do it. And so it's possible. And what's John teaching this semester? I haven't talked to him in a long time. So.
Yeah, he's teaching artificial and biological neural networks right now. So yeah, I'm not really sure if our project is supposed to be focused more on artificial networks or more on biological networks, or if it can be just whatever we want, really.
John is usually pretty flexible. If he hasn't, I mean, of course, he may have changed a lot, but
yeah,
he's a great guy.
Mm-hmm.
I think he's in my old lab space, actually.
Oh yeah.
When I left the physics department, he inherited my space.
Yeah. Huh.
Okay, let's see. Who do we have next? Is it Fanbo?
Yeah. So, hello everyone. My name is Bolson, and I'm right on master in the IC department. And I'm working with Professor Jed Howe, like in the molecular dynamic systems. And I have previous experience like in working in the nano hub, so I'm pretty comfortable in working like a nano hub application. So, but I didn't have any background in the biology. So for my project idea, I was trying to investigate like the similarity and the difference between the simulation, like both in the nanoparticle and for the cells, like in the biology.
Well, the one thing, I mean, of course, the size scale can be an issue, but one thing that you can do, of course, and we'll talk about this a little bit more today, is that the things that we call cells in these simulations are simply volumes of material. And so depending on what properties you attach to that object, that cell object could represent many cells, a volume of fluid, a nanoparticle, a virus, or a single cell or a sub-volume of a cell. And so potentially one could build some simulations which included, again, the length scales tend to be a little bit fierce with nanoparticles because they're so small. But if you're willing to accept that your nanoparticles are spatially a little bigger than they are in real life, one could put nanoparticles and have them interact with cells in a simulation framework like this. Andy's simulation environment also is pretty good, would be good for that. But we'd have to see how much of the pieces you need for it are available. But that's great.
Yep, thank you.
Okay, Callie.
Hi, my name is Callie. I'm a junior, double majoring in ISC and math. And regarding my project, I have a couple routes I'm considering. The first is looking at trying to look at drug therapy in some sort of biological system, whether that's cancer or tissue that's otherwise infected by something. And then the other idea is that I'm interested in potentially looking at the filament modeling for cyanobacteria that Dr. Sumogi, I believe, is working on. I just feel like these are two areas I really haven't looked at yet. I mean, in the modeling work I've done before, I haven't really looked at drug delivery or pharmaceutical treatment, and I haven't looked at cyanobacteria or anything related to spindles. So I'm kind of weighing which one I might want to do more, and I'll probably have another talk with Dr. Sumogi if I think I'm going to go the spindle route.
Great. Okay. Adam.
Hi, my name's Adam. I'm an undergrad majoring in informatics, and I haven't really thought about a possible project idea yet.
What's your general area of interest? Anything that appeals to you? You don't have to. You can find somebody who has a strong idea and tag along with them too. It works. But I just any sort of ideas about what suits your taste to begin with? We're not committed to anything by saying that.
I don't really have a strong background in biology, but I have a friend that after this class is gonna like teach me like the basics and stuff, what I need to know, and then after that, I'll probably decide what I'm gonna do.
So some years this course is more, as I say, this course adapts to a lot to the students. And so some years it tends to be more compute heavy, some years it tends to be more math heavy, some years it tends to be more bio heavy. And so we'll try to adjust it as we move on. And you're free to make steering corrections too and requests.
Okay.
All right. Okay, let's see. Furkhan.
Hello everyone, I am Furkhan. I am a PhD student in Intelligent Systems Engineering. My major was bioengineering, and then I continue in my master with in the Systems Biology and Bioinformatics program. And actually, I am retaking this course, and so maybe I thought I would like to work on the work with Mechanica, I guess, because it is a new benchmark to work on it, and also I'm too excited because it is in Python, and I want to be experienced with it, and yeah. And as you mentioned, maybe I would go for the zebrafish epibole project. Might be a good choice actually.
Okay, we'll have to coordinate that a bit because a number of people seem interested in Mechanica, and so we'll have to see. I mean, we'll have to see how much we can rope Andy into giving some extra lectures. It would be great for him in terms of developing the software and teaching it, but I can sit in the audience and learn how to use it just like you. So that would be great. And you'll have to, Furkhan, you'll just have to endure the repetitions, I'm afraid. And you can correct my mistakes and maybe help people out where you got, there was places you got stuck before, you can help people out, okay? Appreciate that.
Sure.
Okay, and let's see, is it Simpla?
Hello, I'm Simpla. I work in the Department of Biology. and I study the ultrastructure of Drosophila eye and do comparative study of the eye of Daphnia, Tribolium, and Drosophila, and trying to see how the evolution has worked to, like these three different organisms have different eye structure. So my idea of taking this course is to see what kind of techniques or technologies available, because I'm very focused on biology and have no idea about anything else. And we have been doing a lot of genetic research and all those things, using genetic tools and all those things. Lately, I've been trying to do some modeling using the Amira software, in which we take some images, like serial sections of images, and stack them together and try to develop a 3D model. So my idea of taking this course is to understand what other techniques are available. Is there something available that I can enhance my research, and develop data in a different way to present and try to be more technical kind of thing. And so, yeah, I attended the last class, looked at things, and I've not yet decided what I want to do, because I'm not able to think how any will be related to my research and how much I can actually give an input in any kind of project that I'm doing, because I have no informatics background. Math is long time ago kind of a thing. I've been working for 10 years now, so a little out of date with everything. So I would like to think more about what I want to do and talk to you, Dr. Glazier, and let you know.
That's fine. I mean, I would say that, and people have heard me say it so many times that it gets, maybe it's not even meaningful anymore when you repeat something so many times, that biology is the hard part. Understanding the biology and what it is you want to represent is hard. Once you've done it, it's not that writing computer code is trivial. It's not. But understanding the complexity of biology and being able to organize it and frame it is really hard. And so having somebody, if you're willing to work with, maybe partner up with one of the other students or two of the other students who are more computational in their background, that can be really productive. They can help teach you computing. And maybe you can help teach all of us some of the basic biology that we need to know. You heard one of the students already saying that they wanted to learn biology in this class.
Yeah.
So I hope people can come together and that there can be some synergy, symbiosis in terms of developing ideas. Last year, we had a student who was very biological in orientation who helped us with the COVID models. And he actually gave two of the lectures in the class on the immune system. Very nice, purely biological lectures on the immune system that were extremely helpful to us. So having somebody with a more biological background would be great. Technically, we've wanted for years to be able to do imports of segmented images into CompuCell's initial conditions. So if you're actually interested in that's more of a software problem, but how you take 3D microscopy images and turn them into initial conditions is something that we worked on some. It's a hard problem. I'm talking with a group at Chan Zuckerberg in San Francisco that's doing a lot of that. And it's quite interesting. And so the other thing I was going to say is it's been a long time, and it's probably on a different, it's more on a higher level, larger scale than what you've done. But there are some CompuCell models that have been published a long time ago of Drosophila eye development. And so we might be able to go back and look at some of those and ask a question about whether one could build more meaningful models today than we were able to do 15 or so years ago. The organization of the Ommatidia in Drosophila is a very interesting problem. Those were 2D models, but one could try to think about some of the three-dimensional organization of the Ommatidia.
Yeah, that would be interesting, and that would be relevant to my work. And I'm trying to, try to develop methods of putting data out in a different way rather than just, like some transmission electron microscopy images and all those things. So I will look into it and try to think of something and be in touch with some people to see if they can, or otherwise I'll just tag along into some project and just have some technique knowledge.
Right. I mean, we tend not to do a lot of ultrastructure. So a lot of the world is what you would see through a 20x eyepiece. I mean, a 20x objective. That's sort of the scale of resolution. Simulations like that one of the cell crawling, the single cell crawling, would be more like 60x objective. It's not that often we drill down to the SEM scales, or well, SEM scales, yes, because we used to use SEM quite a bit for some of the experiments we did. We don't usually drill down to TEM scales because we don't usually go inside the cell that much. Our cells maybe have a nuclear compartment, maybe a few other compartments, but the detailed the macromolecular machinery of cells we don't typically represent. But those are things we can think about.
I'm more interested because, like all the genetic manipulations that we do, we are trying to understand the organization of the cells that makes a functional Ommatidia and what kind of gene. So I think, so I'm not interested too much into the ultrastructure. It's more of the membrane movement, which helps in how it is organized, basically how the Ommatidiums are ultrastructurally organized to give a shape. So I think so we'll be okay with the, I mean, we're not into that much of detail, so, but I'll think about more.
Well, that's fine. We could certainly go back and revisit those old papers and see what people have done with CompuCell and other modeling methods to try to understand that. Certainly, insect eye development's a fascinating structural problem. Okay, did I get everybody? So it's a diverse audience, as you see, which is good. Any comments or questions for each other before we go on? There's going to be a little bit of mathematics in this lecture for the people for whom it's meaningful. That's great. If it's not meaningful, don't worry too much. You can either revisit it, or a lot of it is there because I want to be honest and show you what's under the hood. But in fact, you don't need to know the details mostly to do the work that we do. I wouldn't say ignore what's being said, but try to follow it if you can. But if it's too much math, don't worry too much. I'll try for future to reduce the amount of math, but some of these lectures were developed with a slightly more mathy audience probably. Okay, so I want to talk today about CompuCell, and we're going to talk a little bit about how CompuCell actually physically represents cells computationally and what they do. And we'll talk a little bit about actually how you translate those ideas into computer code, which you can then run. And I hope we can get started pretty quickly in writing some CompuCell simulations and running them. Hoping that NanoHub will behave today. So the first thing to say about CompuCell, if you're talking from a biological point of view, as I say, typically the spatial resolution we're working at is what you get with a 20x objective in a microscope, maybe 40x. And so here, you've seen this picture before, are some pictures of some cells. These look like they're epithelial cells, and we can come, for the people who are not biologists, we'll come back and talk a little more about concepts like epithelial versus mesenchymal organization, which are the two basic architectures that organisms use to build living tissues. Even though we're three-dimensional organisms, actually we're mostly composed of sheets of cells. What we really are lots and lots and lots of layers of 2D sheets that are folded up into tubes or layered structures. And then between them is what's called connective tissue, which is truly three-dimensional. But there isn't that much of it. Mostly, even though we look 3D, we're actually composed of a lot of 2Ds that folded up. We're a little bit of, origami is a bad example because paper doesn't fold the way the tissues fold. More like crumpled up cloth, because tissues tend to be soft. In any case, the first thing to know is that CompuCell represents space as a lattice. And for people who've worked with Paul Macklin and Physicell, his is what's called an off-lattice technique. His cells don't live on the lattice, they live freely. We are more like the voxels or pixels in a television, where each point in space corresponds to a point in a grid. And that has some advantages and some disadvantages, which we'll run into pretty quickly. But at least conceptually, it's pretty simple. If I have a picture of cells here on the top, and suppose I want to have this, can people see my cursor?
Yes.
Yes? Okay,
Yes.
good. So if we have this cell here, this approximate hexagonal cell, all of these blue voxels would correspond to that cell. And so there's a one-to-one mapping between the pixels or voxels in the space of spatial simulation and the cells. And so the cells live in what's called a cell lattice, and that's the primary place that things happen in CompuCell. The second thing is that those voxels are just pictures until you assign them properties. And in the real world, things interact and move around because of forces. And if you're a physicist, there are two ways of representing forces. You represent forces directly as vector quantities, forces. But physicists are very fond of energies, in particular potential energies. And so if you remember back to undergraduate physics class, the force on an object is the gradient of its potential energy. And CompuCell can represent explicit forces on surfaces and on objects, but it tends to use what's called an energy-based formalism or an effective energy formalism, where instead of representing forces, you represent the potential energy that would produce the force that you want. And the reason for that is numerical simplicity. A force is a vector quantity, so it has a direction and a magnitude, whereas the potential is a scalar quantity. It simply has a value, either a position or associated with an object. And so it's a simpler construct mathematically, but it does mean that you have to take a derivative every time you want to calculate a force from a potential energy. And what we're going to do when we build simulations is that each of the behaviors or interactions that our cells have what are technically called processes, will correspond to a separate mathematical term in the effective energy functions. So just the way if I have a cup and I push on this cup here, my finger is pushing on the cup and applying a force on the cup, but gravity is simultaneously applying a force downward on the cup, and my other hand, my left hand, is applying an upward force to keep the cup from falling down. And if I jiggle the cup, the tea in the cup is moving in response to the force of gravity, the force from the wall of the cup, and also from the viscous and frictional forces inside of the fluid. The net force on any given particle of fluid or on the cup as a whole is the sum of all the forces acting on it. In the same way, the potential energy on the cup is the sum of all the potential energies that the cup experiences. And so this gives us a way of writing all of the processes, all of the behaviors and interactions that the cell is participating in the form of a simple sum of terms, each one of which represents a distinct physical or biological concept. And it's that ability to add terms that represent different mechanisms independently of each other, sum them together and see what they do, that makes this kind of modeling very powerful. The detailed mathematical forms may look long, and some of the notation may not be so familiar. We can review it. But what you have to think about is basically that each of these is going to, each term in this long-looking equation represents a different biological or physical mechanism that is going to be applied to the cells. You're not probably going to have to worry too much about the mathematical detail of how those mechanisms are implemented. But the idea that you're going to independently add mechanisms and see how they combine is going to be important. And it's called an effective energy because some of the things really are energies. Some of the things are really forces. If I have gravity acting on a cell, that's a real force. On the other hand, if the cell is crawling in response to external chemicals, what's called chemotaxis, the chemicals are not pushing on the cell. Instead, the chemicals are sending a complex signal that's causing the cell to reorganize the motors that live inside of the cell that then push on the cell and cause it to move. So if you're hungry and you follow a gradient of a smell to get to the pie that smells good, that's cooling off in the window, the molecules that are sending you the signal are not pushing on you. But your response in moving towards the source of the signal looks as if the molecules were pushing on you. And so that's what is called an effective energy. It's a behavior that looks like it was caused by an energy, but isn't. And that's something we're going to run into a couple of times during the course of the semester. And that's why you'll see the term effective energy thrown around. The last thing is that if you have forces or energies and you have objects, you have to know how the objects move in response to those forces or energies. In the real world, people will learn in grade school, high school, or college, depending on when you took your first physics course, about Newton's law, that force is equal to mass times acceleration. And that is what you learn in the textbook, and of course it's how the world works at some level. On the other hand, starting from Aristotle on, people didn't believe that that's how movement happened. And the reason they didn't believe that that was how movement happened was because in Newtonian world, if you push on something and start it moving, it will continue to move until some other force stops it. And of course, at the scale of astronomy, planets obey Newtonian movement very well. But in the real world, if I push on this cup and start it moving and then stop pushing on it, it's going to stop very rapidly because of frictional forces. When I deal with smaller and smaller objects, inertial forces, the things that keep planets orbiting the sun, for example, get to be weaker and weaker relative to drag forces or frictional forces, for example, due to the fluid the cell is moving in. And so if you ask, there's a famous calculation you do in a biophysics course, which is you ask if a bacterium is swimming due to its flagellum and it stops spinning its flagellum, how far does the bacterium coast due to its inertia before it stops? And the answer is that it goes less than the diameter of a proton. It effectively stops instantaneously. And so at the scale of individual cells, for the most part, instead of having force and acceleration be proportional to each other, force is proportional to velocity. If I push on something, it moves. If I stop pushing on it, it stops moving instantaneously. And so if I'm doing molecular dynamics, the world is Newtonian. If I'm doing fluid dynamics at large scales, I have a complex mixture of viscosity, which is dissipative, and inertia. And there's something called the Reynolds number, which characterizes that ratio. But at the scales of individual cells, for the most part, inertia is negligible, and things move with a velocity that's proportional to the force. And so once we've defined the forces, that was those long equations at the top of the slide, we need the cells to move, the objects in our world to move, with a velocity that's proportional to the force. And we do that using a stochastic, a probabilistic algorithm, and we'll come back to that in a few minutes. But the result of that is what's called perfect damping, which is that as soon as you stop pushing on something, it stops moving. And so we do have to get used to a world where there's no inertia. And there's some interesting physics paradoxes associated with that, which if we were in a biophysics class, we'd go into. You put those three things together and you get simulations like that one that we saw at the bottom of the vascular tumor. So that's the physical concepts and the mathematical concepts of how CompuCell represents the world. When we're going to build CompuCell simulations, we have to turn those ideas and mathematics into computer code. CompuCell uses two kinds of code to specify models. There is something called CC3D ML, which is a markup language, which is a custom specification of particular aspects of computer simulations, and we'll be working with those today. And then there also are Python scripts, and CompuCell uses Python to do, well, you can do everything in Python. You don't need the XML. XML historically was developed before the Python. Most models will use a combination of the two. We have this editor, Twidit, which we saw last time a little bit of, that allows you to build these simulations. It has auto code generation to help you so you don't have to remember as much Python. Python is a wonderful language, but it builds traps for the unwary and sometimes traps for the wary too. And so it's very helpful to have some boilerplated code to make things a little bit easier. And again, TJ and Josh and I, and Furkhan is an expert, will do our best to help people when there are Python landmines hiding. People who took the class last semester, there's going to be more Python this semester, And it's going to be somewhat different in structure from what we were used to, not enormously so. So the first thing we could do is create a simulation universe. And let's just do that. First, if we imagine there's a universe, we're on a lattice. So if we have to say basically how big is the box in which we're going to do our simulation. And so if I imagine I'm in 3D, our universe, it actually would be nice if we could have a circular or spherical universe, because most of the things that we want to do would be more efficient that way. But computers are not very good at representing spherical lattices. They like parallelepipeds, rectangles. And so we're going to have a universe that's specified by an extent in the x direction and an extent in the y direction, if it's 2D. And if it's 3D, an extent in the z direction. And that's all we have. And as I mentioned, everything is going to be on a lattice. Typically, we have square lattices. We can also have hexagonal lattices. And it winds up that the difference between a square and a hexagonal lattice is not so big, even though when you draw the picture, they look rather different. In fact, a hexagonal lattice is basically a square lattice where I joggle every other row of the lattice back and forth by half a pixel. And so it's actually quite easy to implement hexagonal lattices on a computer. In 2D, 3D, it's a little more complicated. You have hex close packed geometry. But basically, the lattice is determined by who is your nearest neighbor in 2D or 3D. As I say, CompuCell allows you to have square lattices, which is normal, or hexagonal lattices, to Boolean choice when you set up the simulation. And one concept that we're going to run into right away is that the real world is not discretized into voxels. And in particular, if I look at, in reality, if I'm this black point in the center, and I were to swing my a rope or my arm around in a circle, everything within that distance would be something I could touch. In a lattice, depending on the direction I go, the distance to my nearest neighbor is different. And so if I go north, south, east, or west, I have to hop 1 unit to get to my nearest neighbor, the center of my nearest neighbor. If I go northeast, southeast, northeast, or southwest, I have to go 1.5. And so there's a concept of neighborhood where I have my center pixel here in black. I have my first neighbors 1, 1, 1, 1. My second neighbors 2, 2, 2, 2. And you notice those both make squares. If I want to get something that's smoother, more like a circle, I have to go further out. If I go to third neighbors, I'm still a square. If I go to fourth neighbors, I have an octagon. So it looks like a stop sign. That's getting a little rounder. And most of the time that doesn't make a huge difference, but often we will use not first neighbor, but second neighbor or third neighbor interactions. If I go to a hexagon, the world looks rounder even with nearest neighbors. And so it winds up that hexagonal lattices are a little better behaved mathematically than square lattices, but they're a little harder to conceptualize. CompuCell intrinsically, the lattices in CompuCell are always 3D. It's just that that third dimension is sometimes set to be 1 unit, in which case you're 1 voxel thick. So you don't have anything happening in that direction. But it's called CompuCell3D because all simulations are intrinsically 3-dimensional. Okay. One other thing that you're going to have to deal with CompuCell, which probably won't be so meaningful now, but may turn into an issue when you deal with your projects, is that CompuCell doesn't have any intrinsic concept of a unit. It's up to you to say whether one voxel represents 1 micron, 10 nanometers, 1 centimeter, and so on. CompuCell does provide some things for keeping track of that. But in general, what 1 unit of space represents in your simulation is up to you. That gives you a lot of flexibility in terms of resolution, but it means you have to do some bookkeeping on your own to keep track of those things. Okay, we've already seen that CompuCell as software comes in 2 pieces. There's this text editor, Twidit++, and there's Player, which runs the simulation. If you're TJ or Josh, who are very sophisticated, they like to use GitHub and Git associated editors. There's a lot to be said for that. Twidit, I think, is probably easier to start with because it has these predefined code blocks. So it basically knows a lot of the things that you want to do. And so as a learning tool, I think it's very helpful. Again, you'll go back and forth. When we went last semester, and again, I try not to talk too much about last semester since there are many people in the class who didn't take last semester. But last semester, the editor and the running environment were the same. Everything ran in Jupyter notebooks. Here there is a separate tool for running the simulations from the one you use to build them. That's a little bit less convenient. It's a little bit like the difference between writing text in Microsoft Word versus writing text in LaTeX. In LaTeX, you write the specification for the document, and then you have to turn the document into something that's compiled text. One of the things that's nice about Andy's new code is that it's all Python, and that means that you have the advantage that everything lives in the same environment. Here, the core code, the core CompuCell code is in C++, and it's called as a libraries from Python. Okay. So let's actually we're going to create a nothing simulation where we're going to create just a blank environment. And one of the things that CompuCell has is a little tool called Wizard that boilerplates in some of the basic things that you do a lot. And so what I'd ask people to do now is to start up CompuCell in NanoHub. And let's see, this says CC3D main tool. But then when I'm inside of it, it says, let me launch Twidit. So I'd like everybody to launch Twidit if possible. And let's see, where did it go? There it is. And if NanoHub doesn't play nice with Zoom, I may wind up having to switch to my desktop app. I don't know why. Good. There we are. Okay. So does everybody have Twidit up and running? It's going to be boring if you don't, so I'll do a poll and wait for a minute till everybody has it running. And we'll find if people need help, raise a hand, speak up. Josh is on the call, TJ is on the call. If there's a problem, TJ is the current lead developer of CompuCell. So you've got the horse's mouth, so to speak, here for help if you need it. I know he doesn't work too late, so if you've got a question, ask it soon, because he'll have to go to he'll have to leave pretty soon. But Josh will be available. It's possible that, as they say sometimes, your mileage may differ slightly. Some of these slides I've been using for a long time, and so there may be some small details in which where the current version of CompuCell looks slightly different. And if we catch ourselves having slides that need updating, we'll do that. But I hope I don't have any landmines in the slide deck for today. I see about half of the people have got it running. If you don't have it running, is there a problem anybody's having getting CompuCell up? While we're waiting, how many people tried downloading CompuCell for desktop deployment?
I tried downloading it for my computer, but I was told that I'm unable to open it because I don't have an application to open it. I don't know, Mac decided to be weird with me.
Okay, so Macs have put some annoying security features in that make installation a pain. Josh, can you help Callie with her Mac installation?
Do it. I don't know if you want to set up a break.
So I guess the question for today, colleague, is it okay to use the NanoHub version?
Yeah, that's fine.
And then if you email Josh, he can walk you through the install.
Yes, I can use that.
Set up a meeting. We can set up a meeting and talk anytime.
Is anybody else having issues with Mac install? Windows, it'll ask you for a security override. So when you hit the install button on the installer for Windows, it'll pop up Windows saying this isn't trusted, and then you have to hit advanced, and then you have to tell it to do the install. But it used to be easier. But is anybody else having trouble? I mean, again, one of the reasons we've been using the NanoHub tool is that you don't have to install anything locally, but it does have some disadvantages. So, okay. So there's still 2 people who don't have it running. Is there anybody who needs help on this? That's 8. Okay. Okay. So the first thing that we're going to do is show you how to use Wizard. And Wizard, you go up to CC3D Project. And I don't know why CC3D Project pull-down menu migrated from the left-hand one, which is where it would make sense to me, to the third one over. It used to be on the left, but in any case. And then we're going to do new CC3D Project. Okay. And when you do new CC3D Project, you will have a simulation directory. And if you're going to do things inside of NanoHub, you need to make sure that it's pointing to your home directory. It should do that automatically. It shouldn't be pointing to CC3D NanoHub, something like CC3D. It should have home, NanoHub, your name, and then some subdirectory, whatever you like. Okay. And if I hit browse, it should let me see the NanoHub world. And putting it inside of demos should be all right. Or I could put it higher up, create a directory, do what I want with it. Okay. Anybody having any issues with that? The next thing I have to do is I have to, actually don't know what happened with, well, we'll see. I don't know, it should leave it where it is. I hope that it hasn't disappeared on me by hitting cancel. And then we have to give the simulation a name. And especially if you're running it on NanoHub, which is a Linux system, but anywhere, I strongly recommend not putting spaces or special characters into your name. So we could call this simulation empty, I don't know, it's not going to do anything, empty universe. All right. And you're given a choice. You can, used to be you could have XML only. Maciek didn't approve of that. So now it's either XML plus Python or Python only. We're going to typically select the default, which is Python plus XML. So everybody type a name, find your directory, and then hit next. Okay, so I did lose it did lose my directory. So let me just put it in, I'll put it in this untitled folder one. Okay, and hit next. Okay. And yeah, so actually the format here has changed a little bit in this latest version of CompuCell. The next thing we have to do is simply say how big the domain we're going to work in is. And remember that this is in units of voxels, and what a voxel represents in terms of real space is up to you. And so the default here is 256 in the X direction, 256 in the Y direction, and 1 in the Z direction. That is, we're in a two-dimensional world, which is discretized into 256 by 256 pixels. Okay. And we can define whether we have walls, the world ends at the boundary, or whether we have periodic boundary conditions where one end comes back and is connected to the other. Of course, the real world does not have periodic boundary conditions, but computationally and numerically, periodic boundary conditions are extremely convenient. And so we often, although not always, will use periodic boundary conditions. And we can certainly talk more about what those mean. We can change the size of our lattice here by typing. We could make it 128 by 256 by 256 by 128, something like that. We have a choice between square and hexagonal lattice, which we talked about. This pixel copy range or neighbor order is this question about who's next to whom. Are we only the 4 nearest neighbor voxels that are nearest neighbors, or do we include more? Typically, we're going to make that number 3 or 2 or 3, but we have a choice on that. The other things that we got on this menu are initial cell layout. We heard about the issue of digitizing three-dimensional images. CompuCell doesn't have the best, most sophisticated initial condition generators. So it has essentially a couple of possibilities. You can, in Python, specify initial conditions. Then you can write code to draw complicated objects. But CompuCell by default has a concept of a rectangular or parallelepiped volume full of cells, which it calls a rectangular slab, or essentially a sphere, a circle or a sphere of cells, which it calls a blob. And those are its two default, and I can put multiple blobs or multiple rectangles in different places. And they give you a limited amount of customization. I can also read, if I had, for example, a digitized image, I can read that in. There's a simple file format that allows me to read in configurations called PIF, which we're not going to use much at the moment, but you can use. The only other thing that's in this window that might be mysterious is this thing called average membrane fluctuations, which is set to 10. And that's going to become meaningful when we talk about how things move and stochasticity. For the moment, we're going to ignore it. We can change all of these numbers later on, so we're not going to worry too much about that. So does everybody have this entered like this? People can do that. Essentially, you're not going to break anything by changing the numbers, so you can make them anything you want. And we're not actually going to run this simulation. It's not going to do anything. So it's not that important, but it is showing you how to set up the basic world of the simulation. So these are all global properties of the simulation. So we'll hit next. And the next thing that Wizard would ask us to do is list all of the cells that we're going to have. And so in CompuCell, there's a concept of cell type. These are actually generalized cell types. They might represent real cells. They might represent subcomponents of cells like the nucleus or the cytoplasm. They might represent amounts of fluid. They might represent rigid walls or substrate. They could be all sorts of things. In any case, by default, empty space is filled with something called medium. And you don't have any choice about that. That exists. It's one of the few things that CompuCell gives you for free, is that if I don't specify anything else, the world is full of a cell which has no properties, essentially, which is called medium. You'll also see a number here. In the old days, we tended to use numbers to represent cell types. And you'd have to remember cell type number 1, cell type number 2, cell type number 15. These days, it's a little more sophisticated. And if I wanted to type a cell type, I could create a cell type like mesenchyme, and I could add it to my list. Again, I can always add additional cell types or additional things later on. This is just boilerplating in some code. For the moment, we're not going to use the cell. We don't have any cells. We're just building our empty universe. So we'll hit next. We'll skip. The next thing that we might want to do is have chemical fields. We heard about chemical fields before from some of the other students. We might have a chemical field that represented oxygen in the environment, or glucose in the environment, or it could represent electrical charge in the environment. We can have an infinite number, as many chemical fields as we want. They co-occupy space with the cells, and they live on their own lattices. So if we define 128 by 128 by 128 lattices for the cells, then oxygen would live in its own 128 cubed lattice. Glucose would live in its own 128 cubed lattice, and so on. And so we could add as many chemical fields as we want. Again, for the moment, we're not going to build a simulation that's doing that, so we'll just hit skip. And you should get something that looks like this. And this is some of the things that cells and fields normally do. And these would be predefined modules. These are called plugins for the most part. and the terminology for the computing isn't always the most obvious, that do things that cells do. There's a textbook that was written by two of my old collaborators, Gabor Forgax, the one who went to found Organovo, and Stuart Newman, who's a professor at New York Medical College. And they wrote a beautiful book on the biophysics of development. And the very first sentence of the book says the most important things cells do are stick to each other. Because if cells didn't stick to each other, you'd be a bacterium. Because you wouldn't have multicellular life if cells didn't adhere. And so the first thing that we have in our box of cell properties and behaviors is that cells can stick to each other. And they can stick to each other in a whole variety of ways, which is why there are multiple boxes. I mentioned the idea of following a good smell to its source. Both eukaryotic and bacterial cells follow chemical gradients. It's called chemotaxis. Cells can secrete chemicals into their environment or absorb them. We call that secretion. Cells can grow. They can divide, called mitosis. They can die, death. Something that may be a little less obvious that we'll come back to very soon is that in this world that we're building, nothing exists except medium unless we assert it. And so cells don't have a volume unless we say they do. So we're going to have to assert volumes for cells. We may assert surface areas for cells. You may want to push on them in particular ways. That's what external forces are about. Then the other things those next three are things we don't use as often. Connectivity, sometimes you want to enforce certain rules about how cells behave at a micro level. That's not used so much. Elongation, also not used so much. There are certain things that are numerically expensive to keep track of. And so those are options. The center of mass position of the cell, who my neighbors are, that one we typically do use. The shape of the cell, mathematically speaking, is moment of inertia. The position of each of its pixels, where its boundary pixels are. Those are all slightly expensive computations. Probably these days should be turned, now that computers are very fast, those should probably all be turned on all the time by default. And the other two are things that we can neglect for the moment. Again, we're not going to be doing any of these things, so we can just hit next. And then you'll get a box that says configuration complete, and you hit finish. So everybody can hit finish. And you should see that. Does everybody have that? I'll start a poll and make sure that everybody was able to get there. If anybody's having any trouble with that, let me know. If anybody needs help, speak up. Okay, so what you'll see here now in the editor is on the left-hand side, there'll be essentially a tree structure that describes how the simulation is specified. And if I double click on this to open it, I can see a hierarchy. The simulation is called emptyuniverse.cc3D. And each simulation is actually a directory structure and a bunch of files that specify how the simulation is executed, what its spatial configuration is, what the simulation specification of the player visualization is, and all those things. There are three main pieces to it. Some of them are not so easy to see. You have to open them explicitly. But by default, there are three main pieces. There's something called main Python script. And if I go across here at the top, I see empty universe.xml, empty universe.py, and empty universe steppables.py. Okay. The main Python script is not terribly exciting. It essentially keeps a list of all the pieces that go into the simulation. So essentially a directory of resources. And we typically will not edit it by hand. Twit will typically know how to add and eliminate resources from the simulation. Occasionally we'll have to go in and edit it by hand, but we'll try not to. And so even though it's called the main script because it actually orchestrates everything else. But from the point of view of a user, normally you keep your nose out of it. It's not something we have to play with very much. The next thing that you have is this XML script, and that XML script specifies the configuration of the universe, the lattice, and static properties of the system. And we'll walk through that in a second. And then the Python, which we didn't use, creates a placeholder where we can put Python code that will run. And that Python code might be something to create initial conditions. It might be something to plot graphs. It might be something to save files. It might be something to cause cells to make decisions. Most of the complexity of the simulations is going to go in there. And you'll see these terms of steppables. And Python steppables are called steppables. Not a very nice name, but we've had it for 25 years, so it's hard to change at this point. Because each of those is executed once per simulation step or less frequently. At the moment, you can't execute it more frequently. Our current simulation, the only thing that's actually doing anything is this XML. And so let's take a look at the XML together. I've already done this. We've opened up our XML. And this is pretty small. One thing that we may have some issues with is my monitor is extremely high resolution. And so some things, sometimes text gets to be really tiny. And the resizing doesn't always work, especially when we're using Zoom. So let's see if I can actually, Control plus should expand the font. And it worked this time. Can people see that now? Is that visible to people? You should be following along while we're doing this. Was everybody able to switch, see the XML, get it into a font where you can see it? Everybody, Tyler? Anybody having trouble with that? One person says they need more time. So if I look at my code now, the first line, it's XML. So you can see lots of brackets and slashes. XML is just a text format. It doesn't really have any meaning. You could perfectly well have written this code without the slashes and so on. The first line says CompuCell3D. The first line says CompuCell3D. and then the revision number, and then version number. And the version of CompuCell that's running currently on NanoHub is 4.2.2. And there's just been a new release of CompuCell. So if you're running desktop, it'll be a slightly later version than that. And we'll do our best to get the NanoHub CompuCell updated to the latest release. The differences between 4.x are not that big, so everything should be okay. If you look in my slide, you'll see version 3.7.5. Version 3 to version 4 moved from Python 2 to Python 3. And so that was a fairly major variation. And we may occasionally see an old slide where you'll see some Python 2 syntax that I didn't catch. The main difference between Python 2 and Python 3 from our point of view is that for some reason in Python 3, they decided that print statements must have parentheses around them. And so the old-fashioned Python 2 print statements with no parentheses crashed Python. It's no idea why they decided that was so important to create New World for, but they did. And why they made it so it broke backward compatibility. But that was a Python issue, not a CompuCell issue. All right. So that's the first line. The next code is a box called metadata. And that metadata, CompuCell intrinsically is parallel. And that parallelization is done automatically. But the more you push the code to be parallel, the more there's some small, I wouldn't say mines hiding in the parallelization, but there's some divots or sort of water traps hiding the parallelization. And so for the moment, when simulations don't have to run fast, we'll turn it off. But if you have a modern CPU, my laptop, for example, has 8 cores, 8 physical cores. And so if you're on a modern laptop and running locally, and you have a bunch of cores, if you set the number of processors equal to twice the number of physical cores, the code will run a lot faster. So if you have 8 physical cores, you set the number of processors to 16, it'll run about 8 times faster. On NanoHub, I don't know whether we have access to multi-threading, so I don't know what would happen. It shouldn't do anything bad if you don't have them. Then we have a block that's, you'll notice XML, you always have a start tag, metadata, and an end tag with a backslash, with a slash, and metadata. So those tag pairs are very typical of XML. It's also possible to put the end inside of a bracket like that instead of doing a tag pair. And so you'll see the next block is called the POTS block. We'll find out why it's called. There's certain things that have names that are historical that I wouldn't have picked these days, like steppable. POTS is because the basic algorithm that we're going to use to move cells around is derived from a physics model called the POTS model. And this is where the lattice structure is defined. And the basic line that we cared about is creating our lattice, which is line 14. And it says the dimensions of the lattice are 256 by 256 by 128. I can go in, and if I wanted to make this 1, I can edit it. So I can make it anything I want. Steps is how long the simulation runs by default. Very often I will let the simulation essentially have an infinite length and have some condition that I check to make it stop, or I'll interrupt it by hand. Sometimes I'll want to force the simulation to stop at a particular time. That fluctuation amplitude that we talked about has the unfortunate name of temperature, and it does not mean the room temperature. It's not 37 degrees or 27 degrees centigrade. It has to do with the fluctuation amplitude of the cell membranes. And we won't play with that until later. Neighbor order, this lattice architecture that we talked about, whether a lattice is periodic or not in our X, Y, and Z directions, which are controlled separately. And then, and we'll come back to this in a little bit, but we can walk through the code, just walk through it, and we'll come back to that. But we didn't create any cells. We didn't create any cells in this simulation, just an empty universe. Plugin name cell type defines the cell types that we're going to use. And those have to be listed. So if we're going to have a cell called mesenchyme, it has to be listed here. In this case, we didn't create anything. And as I mentioned, there's always this default thing, which is medium, because the cell lattice has to have something in it. And if we have nothing in it, that nothing is called medium. Imagining fluid medium, water, or maybe air. It happened that by default, tracking the center of mass was turned on. So there's the center of mass plugged in. And it also happened that by default, it actually laid out a bunch of cells using that box initializer, that slab initializer. Although, since there are no cell types to initialize, it can't actually put anything in. All right. So now that we have our universe, which is just a rectangular box, maybe with periodic boundary conditions, and this slightly strange idea of neighborhood. Biology tends to be fuzzy. Boundaries in biology are fuzzy, so the idea of having neighborhood not just be nearest neighbor, but slightly further away is not a crazy one. It corresponds in some sense to the fuzziness of boundaries in biological systems. So we're going to put a cell in this universe. And I skipped a little bit of biological background, and so I'll use some biological terms. If those terms are unfamiliar, you can ask for definition. I often say, and I think you'll hear me say it multiple times, most of the level of knowledge that we need of biology and physics and mathematics is at the level of Wikipedia. Sometimes we need more than that. Sometimes Wikipedia is too detailed. But in general, Wikipedia is a pretty good ballpark for the level of detail. Just the way, if we think about using a microscope, it's a 20x microscope, which is sort of a workhorse, moderate resolution microscope. When we're going to talk about scientific concepts, most of the time we need basic correct information, but not a huge amount of detail. And so if there's a term that you see in a talk that you don't recognize, I strongly recommend Wikipedia-ing it and reading quickly the basic introduction for that concept. Sometimes you need to go in a little more detail. Occasionally, as I say, Wikipedia will be too much detail, but in general, it's written at about the right level of detail to start with. And so cells, and I have a whole deck on this. In some years, I would spend a whole lecture on the biology of what cells do and how they look. This year, because we have fewer lectures, I am hoping that people can look that up a little bit on their own. The simplest cell that you could think about is basically, cells can be very complicated. Neuron has a lot of very complicated structure and a lot of very complicated function. Some cells are, epithelial cells are polarized. They have a front and a back, a top and a bottom. There are also other cells that are what are called mesenchymal, which are more three-dimensional and aren't, they can also have orientation, by the way. They can be polarized as well. But we'll imagine our simplest conceptual cell is something like an amoeba, which is a blob of living material that doesn't intrinsically have a front or a back, top or a bottom. It's more or less isotropic blob. You could think of it as maybe an underinflated beach ball with a nucleus that's alive and moving actively. And that would be what is called a mesenchymal cell. Mesenchymal means it doesn't have that orientation that we will come back to for epithelial sheets. And it's unpolarized. And if we're going to put a cell somewhere, it has to have a location. So it has a position, and it has a volume. And one of the things we're going to have to get used to in this modeling world is that nothing exists unless we assert it. So a cell doesn't have a position or a volume until we say it does. And so we're going to have to, at the very minimum, create an object, which is a cell. It's going to be given a type. We already had this concept of cell type. So every cell has to be associated with a type. Here I called it mesenchymal. We're going to attach a volume to the cell. And it's going to have a position. And if I want to do that, now I have to dive in a little bit to how CompuCell represents cells to understand what's going on. I mentioned that everything lives on a lattice. And CompuCell represents cells using a formalism that goes back to this physics-based Potts model formalism, which was originally developed in the 1950s. And then work that I did back in the 1980s and 90s. So anything that is volume excluding in real space that we want to represent as material objects, which live in the cell lattice, is going to be called a generalized cell. And a generalized cell is simply a collection of voxels that are associated with that object. They might be connected to each other. They might be separated from each other. But here I have a rectangular grid. I don't know why that line is invisible in this pasted, but and I see white, which corresponds to medium, where I haven't defined a cell. And then I have defined three cells: a yellow cell, which occupies these pixels, a red cell, which occupies these 10 pixels, and a bigger green cell at the bottom. And so this is about as simple a representation of an object as you could have. You imagine pointing a video camera at it, every voxel or pixel of the image that corresponds in the position of that object, you assign to that object. Okay. One of the things that we're going to have to deal with, which is a little bit counterintuitive, is that while the volume of a cell is defined, that is, this pixel is part of the green cell, this pixel is part of the red cell, this pixel is part of the yellow cell, the surfaces of cells are not defined. The surface is what's called a fiat object. It's where two voxels of different cell that belong to different cells are next to each other. And so the boundaries here are indicated by this arrow. This edge here between white and red is a boundary. This edge here between yellow and green is a boundary, and so on. But while the volume has a physical reality in the model, edges don't. If you're working with a center model, which is how Paul's physicell works, cells have positions but have neither volumes nor surfaces. So the idea that you could have a volume and no surface from a computational point of view isn't as strange as it might sound at first. So that means since cells consist of a collection of voxels, that automatically any cell has a volume. It doesn't automatically have a surface area, but it has a volume. The surface area will depend on that interaction distance that I specified earlier. Okay. One of the things that we're going to have to get used to, and this is going to be true of any kind of modeling of cells or physical reality for that matter, is that certain things that are properties of material objects that we're used to in the real world are emergent in some way in a computation. And so let me give you an example in the real world. Suppose that I have a balloon and I want to know what the volume of the balloon is. Well, the volume of the balloon depends on how much gas, how many moles or how many kilograms of gas I put into the balloon. It will depend on the temperature of the balloon. It will depend on the pressure outside of the balloon. And it will depend on the elasticity of the rubber on the surface of the balloon. So while the amount of material inside of the balloon, the amount of gas in moles or kilograms inside the balloon is well defined, the volume of the balloon will depend on the ideal gas law, which depends on the temperature and the pressure. And so the mass of the balloon is a well-defined property of the balloon, but its volume is a behavior. A volume is a result of that mass doing what it does under the constitutive relationship, which is the ideal gas law. Therefore, we're going to find that cells in CompuCell behave a little bit like balloons. We can measure the volume of a cell at any given time, but we can't enforce the volume of a cell. So what we will have to do is we will have a lot of things which have a concept of a target volume, and then the cell will try to have that volume, but it'll have to compromise. As an example, suppose I had a cell and I asserted that the cell had a diameter of 10 microns. And I put two cells next to each other with their centers 1 micron apart. They overlap. Well, real world objects have volume exclusion, so I can't put two rigid spheres in a situation where they overlap. I can't simultaneously specify the position of objects and their size freely. And so we're going to find that a lot of these things, we'll set our targets and then constraints, and the system will have to find a way of trying to do what we've asked it to do, given those constraints and the reality of the other things in the world. If there were only one cell in the universe, we could give it any size we wanted. But if there are multiple cells, they're competing for space, and that competition and who wins and how the volumes are allocated will depend on their material properties. So think about the example of a balloon. If I say I've put 1 mole of gas into a balloon, how big is it? I can't answer it if I don't know the pressure outside the balloon, the temperature at which the balloon is, and the material property of the membrane. Does that make any sense at all? So we're going to find that the volume of a cell is an emergent property. However, the quantity that corresponds to the mass, the amount of gas we put into the balloon, is something that we're going to call the target volume. And the quantity that would correspond to the compressibility of the gas, or the inverse compressibility of the gas in the ideal gas law, is something that we're going to call the lambda volume, which is the strength of the volume constraint. And this is something that if you've, actually it's not, physics should teach it, and it tends not to. It's actually true of all physical objects. It's not a difficult concept, but if you're not used to thinking about things in that way, it takes a little bit of getting used to. So we're going to imagine that we want a cell to have a given volume. And we're going to assign that cell a target volume, which corresponds to what we want the volume to be. The bigger the target volume, the bigger the cell will be. Smaller the target volume, the smaller the cell will be. But there's no guarantee that the actual volume of the cell will be the same as the target volume. It might be bigger or less. And that's the thing that is going to push the cell to have that volume is a constitutive relation, if you're a physicist, or a constraint, if you're a mathematician. And the classic example of a constraint is a spring. If I have, see, I usually have, do I have a rubber band? Here's a rubber band. Can people see my screen? Here's a rubber band. If I have this rubber band and I hold it loose, so I'm not pulling on it, it has some length. If I pull on it, it stretches and it gets longer, but it applies a force to pull back. And more or less, the force that it applies is proportional to the stretch. If it's behaving like a spring, an ideal spring, Hooke's law spring. In the case of a rubber band, in the case of a real spring, an ideal spring, if I could try to compress it, it would push back. In reality, a rubber band doesn't push back when I try to compress it. It'll just crumple up. If I have a balloon full of gas and it's at its equilibrium, there's no net force inward or outward, and I try to make it smaller, I have to squeeze it. So I have to do work on it. If I try to make it bigger and pull on it, I also have to do work on it. So a balloon is a three-dimensional analog of a spring in two dimensions. Okay. When the actual volume of the balloon equals its target volume, there's no net force inward or outward. We say that constraint on the volume is satisfied. The same thing with this rubber band. If the rubber band, my fingers are at the distance the rubber band wants to be, they're not pulling on my fingers and my fingers aren't pulling on the rubber band. If I stretch it, the rubber band pulls inwards. And if it were an ideal spring, if I pushed it in, it would push out on my fingers. So the constraint is satisfied when there's no net force. If you've done optimization, say in a data science course, or in any other kind of course, then you'll be used to the idea of a loss function or a penalty function, and the idea that that at optimum, those things are minimized. So what is a constraint function? A constraint function, it could have all sorts of different shapes, and it could apply in all sorts of different conceptual worlds. But a constraint function has a particular property, which is that if there's some value associated with an object, and there's a target value, when that object has the target value of when the object property is equal to the target property, the difference between the value of the object property and the value of the target property is 0. So again, if I have my rubber band, and the length of the rubber band is its target length, then the force on the rubber band is 0. If I stretch it, the force goes up. And so what I want is something where the constraint function gets bigger as I move away from my target. And that means that my constraint will have the property that it pushes the configuration of my system, whatever it is, to satisfy the constraint. And you can imagine a mass on a spring doing that. You can imagine a ball rolling in a bowl, trying to find the bottom of the bowl. Those would be classic examples of constraints. But the key thing is you want this function to get bigger as you go away from whatever your target is, and you want it to be smooth. Typically, we'll use a quadratic distance, which is what you get for a spring. So if a spring has equilibrium length L, the force applied by the spring is x minus L, 2x minus. And the energy associated with the spring, the potential energy, is the spring constant times x minus L quantity squared. Depending on your convention, there may be a half or a 2 hiding in there, but the scaling is right. And the nice thing about a quadratic potential energy is that if I'm too big, it pushes me back. If I'm too small, it pushes me back. And so most of the behaviors that we're going to assert in our modeling world will be in the form of quadratic constraints. And physicists often use the word harmonic instead of quadratic. And the nice thing about a harmonic or quadratic constraint is that there is a restoring force, which is linear in how far away I am from my target, that pushes me back to my target. And in the case of a spring, it's very obvious that the direction of the force is the direction in which I'm elongating the spring. But these could be very abstract quantities. It could be that I want my cell to be an ellipse rather than a circle. And if the cell is round, there's a penalty. If the cell is too elongated, it's a penalty. If the cell is triangular, there's a penalty. And so there's a penalty, which is will create a force that pushes my cell to be more circular. So I could imagine almost any arbitrary set of constraints I want. If I want my cell to move 3 steps to the left and 2 steps to the right and 1 step up, I could create a penalty function that does exactly 0 when those things happen and gets bigger as I go further away from that. And so it's a very general concept, this idea of a constraint. And again, if you write energies in the form of, if you write constraints in the form of these potential energies, the system automatically generates the forces necessary to satisfy the constraint. If you use a force-based formalism, you have to figure out what the forces are. If you use an energy-based formalism, the way CompuCell does, the physics of the problem and the mathematics of the problem automatically figure out what needs to be done. You don't have to solve it. And so that's the key idea of a constraint. And so in CompuCell, and again, the math we don't have to worry about too much, but I'm going to show it for the people who know the math or the physics. Almost everything we do is going to be written in the form of a constraint. And we'll use this curly H It's another physicist's convention for the effective energy. And for each property or behavior that we want the cell to have, and there'll be some exceptions and I'll tell you about them, we write a sum over the cells of a constraint strength, lambda, that would be something like the spring constant, the Young's modulus of a material, the inverse compressibility of a gas, times whatever metric we've chosen to constrain, the value of some property of the object, minus the equivalent value of the target property of the object. We want the cell to have a volume of 100 cubic microns. Then the value would be the volume of the cell. The target would be 100 cubic microns. And that difference is squared. That creates a force, a restoring force, pushing us towards satisfying the constraint, minus 2 times lambda, that constraint strength, times the difference. So as we move away from satisfying the constraint, the restoring force increases linearly, and the strength of that restoring force is given by lambda. Lambda is big, I'm pushed back very hard, and the constraint is what's called stiff. If lambda is small, I'm allowed to deviate a lot. With my rubber band, if my rubber band were very stiff, I'd have to pull very hard on it to stretch it. If the rubber band were very soft, it would stretch very easily. And so the actual length of the rubber band is a function of the force that I apply and the material property of the rubber band, which is its elasticity, which is lambda. As I mentioned, we've talked about length, we've talked about position, but these could be very abstract concepts. And it's important to remember that any object property or behavior can be written in the form of a constraint. Okay. The nice thing about constraints is that every object is independently trying to satisfy its constraints. But when you put a lot of objects together, they have to compromise. And so if I put a lot of people in a crowded room, you might want to take up a certain amount of floor space. And if the density of people is low, you can do that. But as the density of people in the room gets higher, you get squeezed in and you get compressed. And so the amount of space you occupy depends on how much space you want and how much you're squeezed by others. And again, the final result is satisfied automatically by the system, by your compressibility and the compressibility of the people around you and how many people you're competing for space with. The reason this is important is that the cells will try, in your universe, will try to do a lot of things at once. You might say that they have a target volume of such and such. They might have a target surface area of something else. Depending on the ratio of the target volume and the target surface area, you might have something like an underinflated beach ball, where the target surface area is large compared to the target volume. Or you might have something like a basketball that's blown up to be very rigid and it's under high pressure. Those are both examples of a membrane surrounding a gas, but the target volume and the target surface area are different. And that gives you material, in one case, that's very floppy and compressible. In the other case, it's practically rigid. That ability to combine mechanisms, physical mechanisms, is very powerful. We're going to impose a dynamics that tries to balance all of those constraints. We can never satisfy them all simultaneously, but the system will do its best to satisfy them as well as it can. The bigger that lambda in the constraint is, the more important it balances that constraint against the other ones that it has to distance it. Okay. The most important surface constraints that we're going to run into, and the first one is one we're going to do all the time, is volume. Because if our cells don't have an asserted volume, they're going to shrink and disappear. They'll just go poof and evaporate. The second one that we do a lot is surface area, the membrane area of a cell. So the volume, the target volume, would essentially correspond to the mass of the cell, and the surface area would correspond to the amount of membrane. I'll point out that even with real cells, both of those things are not absolutely fixed. If I squeeze on a cell and put it under high hydrostatic pressure, the cell will shrink. If I put the cell in a situation where it's pulling water in and the osmotic pressure inside the cell is higher than outside, the cell will swell up. And actually, when cells get sick, they swell because they have pumps that are continually adjusting the pressure and the osmotic concentrations. Similarly, with the cell membrane, if the cell volume is small compared to its surface area, the membrane of the cell will be wrinkly or floppy. If the cell swells up, the cell membrane will stretch and get smooth, just the way when I blow up a beach ball, initially it's floppy. As I blow it more and more, the membrane will begin to stretch and I'll get a spherical cell. That actually is something you see when you interfere with mitochondrial function. Cells ATP get depleted. The pumps that pump water out of the cell stop working. Water pours into the cell swells up, and if the cell gets sick enough, it'll burst. The membrane will burst. So these are things that actually correspond to real biological situations. So volume constraint. We've talked about this idea of constraints in the abstract. Volume constraint is very simple. For every cell, I measure the volume of that cell. I have a target volume of that cell. I take the difference and square it. So if the actual volume of the cell is equal to its target volume, that's 0. If the cell is bigger than its target volume, it's a number bigger than 1. If the cell is less than its target volume, it's a number less than 0, but I square it so it's still positive. And I multiply it by lambda volume, which is the inverse compressibility. It's what's called the Young's modulus for the material. So the cell really is behaving like a balloon. It's like behaving like a bag of gas. The pressure inside the cell is 2, really a 2 missing here, times lambda cell times the difference between the actual volume and the target volume. It's actually a pretty good approximation to osmotic pressure, although the lambda volume, the lambdas we'll use will be much smaller than those for water. Most cells also have a membrane area, although the membrane surface area tends to be more adjustable for cells than the volume, and it'll have the same form. Elasticity of the membrane times the actual surface area of the cell minus the target surface area quantity squared. If the volume to surface ratio with the right exponents is small, then the cell is floppy. If the volume to surface area is big, I have something like a basketball that's inflated. And again, that's something I see in real life with cells. Plant cells are under very, very strong hydrostatic pressure that gives rigidity to plants. Animal cells tend to be under low pressure, and they tend to be pretty floppy. There's some exceptions on both sides. Okay. So now, when we come back to our picture of cells again, we can think about a little bit more what's going on. The first thing is that computationally, we have to know which cell we're talking about. And CompuCell will assign each cell a unique identifier using the script Greek sigma as the index. So there'll be a cell number 1, a cell number 2, a cell number 3, a cell number 4, a cell number 5. You're almost never going to care about what that number is. It's arbitrary, but you do need to have every cell have a unique identifier. You have to say cell number 66 is not the same as cell number 99. And CompuCell will, when you create cells, automatically give each one a number. As you add them in, it will add up higher numbers. And CompuCell will tell you how many cells are active at a given time. Since each cell occupies some number of voxels, automatically every cell has a volume. It's how many voxels are inside. So this red one, again, had 10 voxels, has a volume of 10. If I sum how many mismatched neighbors I have, that's the surface area of the cell. And that's going to depend on the rule I use to define how many adjacent neighbors I have for each voxel. And so as I change that interaction length, the surface area of the cell will change. The volume won't. It's independent of the lattice. I also have a cell type. And that's an abstract quantity, but it's convenient for all cells of a given type because different cells, what does a cell type mean? A cell type is a concept of cells that are similar in some set of ways. They're not identical to each other. It's not quantum mechanics, but they're similar. And so having cells that are mesenchymal, having cells that are neurons, I'll have to decide in my model what cell types I want, but I have each one of those named or numbered. And by convention, that's given by tau for T for type. It's also possible to do more complicated things. I could divide my cell into sub-blobs and say that it has a compartment that represents the nucleus, a compartment that represents mitochondria, and so on. So there's possible to have subcells. We're not going to talk about that for a while till later in the class. And again, almost all the properties of the cell are going to be represented in terms of these effective energies. Terminology, and it's not very elegant. We've run into this word steppable and not really talked about what it means. There's going to be a term for effective energies. They're going to be called plugins. And that's a very computery term. And it's because the software actually loads as plugins C++ modules that implement each one of these energy functions. So when you see here the word plugin, it corresponds to one of these energy functions. Okay. Now, we've defined a set of energies and forces. We have to have our cells move in response to those forces. At the moment, the forces exist, but we haven't defined the dynamics, how the world changes in response to these constraints. As I mentioned before, at the very beginning, in the macroscopic world, we're used to things with inertia that obey Newton's law. At the micro level, drag is very, very important. And so instead of F equals ma, we get F equals drag times velocity. And we need to have some way of numerically solving that. And there are a whole variety of different ways to do it. The way it's done in Physicell is one implementation. The way it's done in Mechanica is another implementation. One of the things that's special about CompuCell and these POTS-derived models is that they go back to something called a Metropolis algorithm, which was developed in the 1940s in the context of the atom bomb project, actually. And it was originally developed for solving complicated statistical mechanics problems. It wasn't ever designed to do dynamics, as it happens. The physicists may appreciate that. It's not for anybody. But we're going to have to imagine, in order to understand how things move at the cellular level, we have to understand a little bit of why they move. And some of the movement of cells is passive. If I push on a cell, it moves because it's being pushed. But cells also move actively. The reason they're alive is because they have active movement. And in particular, cells have inside of them motors, molecular motors. And those motors are continually pushing the cell membrane back and forth. When you've heard of amoeboid movement, I should probably, I had an old version of this slide that had a YouTube video of an amoeba. The membranes of cells are continually ruffling due to actin polymerization, depolymerization. And so that membrane of the cell is continually being moved back and forth. And we're going to abstract that fluctuation of the membrane as a thermal fluctuation, as if it were due to temperature. And the reason that that term was called temperature in our model definition was because that fluctuation of the membrane that allows the cell to explore its environment and find where it can move to reduce, to satisfy its constraints, reduce its penalties, corresponds in a physics way to this abstract concept of temperature. In some models, the movement of the cells is deterministic. You solve differential equations to calculate the forces directly, and then you move things in response to the forces. It winds up that that's numerically unstable. And so actually doing those solutions is expensive. And if you work with Andy, you'll discover quite how expensive they are. They leverage molecular dynamics simulations that have developed those kind of solvers to very high art. We're going to do it a little bit differently. CompuCell does a little bit differently. It does things in a less fancy way, but that's numerically very stable. And it's stochastic. And what we're going to do inside of CompuCell is we're going to look at the configuration. We're going to look at the configuration of our lattice. And here I've drawn a picture where I have 4 cells, a gray cell, green, red, and yellow. And this is what happens. And the CompuCell simulations do this not just millions of times, but not even billions of times, but tens of billions of times in a simulation. I pick one spot in my lattice at the boundary of a cell. And I ask the question, if this cell, say this green cell here, pushed the red cell out of the way by one lattice site, what happens to the effective energy? Am I getting closer to satisfying my constraints, or am I getting further away from satisfying my constraints? Now, if I have multiple constraints, some will get closer to being satisfied, in which case I win. Some will get further away, in which case I lose. And I add up all of those differences. Do I get a win here, a win here, a lose here, a lose here? And I add it all up. And it winds up that that net difference is precisely the force that the universe is applying on that boundary. So if the net force is, if my move reduces the energy of the system, the net force is in favor of making that movement. If the movement makes the energy of the system go up, there's a force against me. I'm pushing against that force. And so what I do is I propose a move. I calculate what the energy after the move is, compare it to what was happened before. If the energy goes down, if I win, it's like rolling downhill, I do it. You could say, well, that's the end of it. Nothing rolls uphill. But if I'm moving actively, if I'm passive, if I'm a ball that's a dead object and I'm on a slope, I can only roll downhill. But if I'm a living organism, I have motors and I can bicycle uphill as well, or walk uphill as well as downhill. And those fluctuations exist to allow me to go uphill. And it winds up that ability to go uphill is critical. And it's really what makes living organisms behave differently from dead organisms. It's the fact that I can move against the forces that are being applied to me. Now, exactly how you calculate the probability of going uphill is something that we could go into in more detail another time. Suffice it to say that there is a function which says, if I'm going downhill, I always make the move. If I'm going uphill, I make the move with a probability that goes down as a function of the force that's opposing me. If something is pushing it against me to be very weakly, I'm likely to push back. If I've got very strong opposition, my probability of fighting it is lower. It happens to be called the Boltzmann factor. And again, that comes from physics, it's the 19th century physics. And the scale of energy that I can fight is given by that fluctuation amplitude, T. And I should point out that if you've done any molecular biology and you've looked at DNA, you know that the binding of bases in DNA is comparable to the thermal fluctuation amplitude, comparable to about 1/3 kT, which means DNA, the double helix, is continually unzipping and rezipping. And the fact that DNA is easy to unzip because the binding energies are comparable to thermal fluctuations is what allows things like RNA polymerase, DNA polymerase to work. So again, this kind of kickback and ability to go up energy gradients is something that happens in biology. It's not something that's invented for the purpose of the simulation. So if I imagine I start out with a configuration here where I have cell 1 over here and cell 2 over here, and cell number 2 decides it's going to try to overwrite cell number 1 here, I calculate the energy before and afterwards, and I get an energy difference. And suppose I make that move. I can imagine at the next time that I undo that move, going back the other way. If one direction was energetically favorable, the other direction is always not energetically favorable. So the probability of going one direction is always 1. The probability of going the other direction is that Boltzmann factor, which happens to be e to the minus the energy penalty divided by t. And so the net probability is the difference between going forward and going backward. And that rate is the net rate at which my system approaches satisfying its constraints. If the typical fluctuation energy divided by that, the typical energy difference divided by that fluctuation amplitude is small, I discover that the velocity is proportional to the force. And that's the fundamental mathematics. Again, I don't want to do too much math. The fundamental mathematics that justifies this algorithm. It means that with this funny probabilistic movement of boundaries, my system moves smoothly with fluctuations, but on average moves as if the force, the velocity is proportional to the force. All right. And that's the final result. Now there's some constraints. If the forces get to be very big compared to the fluctuation amplitude, doesn't work. And so we have to watch that. That's why it's an adjustable program. This has some funny properties because only one point on the lattice is moving at any given time. Things move not simultaneously, but sequentially through the lattice. So I have to pick my points at random to make sure I'm not biased. That means that the method in principle seems slow, although the computations individually are quite fast. But it also means that the method is absolutely numerically stable. And if people have done mechanics calculations with finite element methods, so they know that that's not an obvious thing for mechanics calculations. And so this is it seems rather abstract and arbitrary perhaps, but actually, again, this method was developed back in the 40s to try to solve very, very hard numerical problems related to atom bombs. When in those days, the computations were still being done by hand. So numerical convergence was actually a big deal. It's sort of an accident that method works so well here. All right. Because you do one pixel at a time, one voxel at a time, that sets a time scale for the speed at which things can move. There's a unit called the Monte Carlo step, which is trying to update each site on the lattice once. And that's my fundamental time unit. And just as I can define the spatial coordinate to be anything I want, I can make one voxel correspond to a micron, 5 microns, half a micron. I can define my Monte Carlo step to correspond to a second, a minute, an hour, with the caveat that nothing can move faster than one voxel per Monte Carlo step. And so, depending on what timescales I have in my real problem, if the cell moves at 1 micron per minute and the voxel size is 1 micron, then I can't make my time step be bigger than 1 minute. And so that's something we have to learn how to play with. Again, when we're doing abstract calculations, it won't matter. But when you're converting real problems into CompuCell, you have to think about that. Computationally, CompuCell has two fundamental modes. It will do all of its plugins, all of its energy functionals, sweeping over all of the lattice, trying to update each voxel once. And that's called the Monte Carlo step. And then it will look at its Python code and run whatever Python code it has specified to do things like cell division, cell death, cell signaling, and so on. And that alternation between relaxing the configuration of the cell lattice and running the stepables is why they're called stepables, is the basic rhythm of CompuCell. It will move the cells around a little bit. It will then adjust everything else. It will then move the cells around a little bit, adjust everything else, and so on. And so that's how CompuCell sequences time. That everything else could be things like diffusion of chemicals in the environment. It could be subcellular networks updating their states, and so on. All right. So now let's actually do this in the time we have left. So let's create a simulation with a single cell. We're going to give it a target volume of 1,000 voxels. We'll imagine that the lattice constant is a micron. And so that would mean our cell is about 10 microns by 10 microns by 10 microns, which is about typical for your average mammalian cell. Okay? So let's do that. We're going to come back to here. we can get rid of everybody come back to your CC3D project. We can get rid of the one we've been working with. So we can just discard it. Okay. And now we will go back and go CC3D project, new project. And let's call, this is called new simulation is all right. We could call it, oh, I don't know. Well, this is supposed to be leading up to cell sorting. So let's just say cell volume. You can call it something that you like. Okay, everybody create that. If anybody needs help, just raise your hand. Because there's still a couple of people who haven't done this yet. So we'll create our simulation. We'll call it cell volume. And again, the size of the lattice doesn't really matter for the purpose of this demonstration. We can leave it at 256, 256, 1. We'll keep it square lattice. Let's change the pixel copy range to 2. And instead of using a rectangular slab, we'll use blob initializer. So click the blob button there. Everybody got that? We'll hit next. And now we have to create a cell. So we're going to type something. We'll type. The cells happen to be mesenchymal, so let's call it a mesenchymal cell. We type the name here, and we hit add. You'll notice it shows up on our list here. So try that. You can, if mesenchymal is too long, you can call it cell 1. Doesn't matter what the name is. Don't call it medium. You can't reuse a name. But beyond that, you're pretty much free to call it anything you want. So we'll hit next. We don't need a chemical field, so we skip that one. And this time, we're going to give the cell a volume and a surface area. Remember, it didn't have them until we asserted it. So we call volume flex and surface flex. And that means that we're going to define the volume and the surface area of the cell. So click those two buttons. Next, finish. Okay. And now I'll see something like this. Let me double click on this so I can see my code. The Python won't have anything in it. I can look at my XML. And you'll notice that now I have my cell type here, mesenchymal cell. I have a volume plugin. And I have a surface plugin. And so now my wizard has created those energy terms that will create constraints to define the volume of the surface. And so now I actually have a simulation. I can go back up to the top here, the name of the simulation, select it here. And if I right click here, I can hit open in player. And I should be able to switch over to player. And I can see my simulation. Can everybody try to do that? If you have trouble with that, let me know. And Furkhan, your patience is appreciated while we get everybody up to speed. Is everybody able to do that? If you can do that, you have now actually written a simulation. Which wasn't so terrible. Now, I can't promise that every simulation you write will be that easy, but it's actually not that much work. You can see I have these little, the color scheme isn't the prettiest. They have yellow outlines and they're red volumes, but I see my little mesenchymal-like cells, and they're fluctuating. You see they're fluctuating, they're crawling around like little amoeba. They stick to each other a tiny bit, not very much. And you'll notice they have a volume, and I didn't specify what the volume should be, but it actually is not unreasonable. The choice of surface and volume that the defaults are, it's not unreasonable. So I want to wait. I'm going to make sure that everybody has this running, because this is sort of the basis. Once you can do this, everything the rest of the course will do will be built off of this. If anybody needs help, let me know. You'll notice, I'll pause this for a second. If I look in my console, this has already gotten to 11,840 time steps. So the simulation runs pretty fast, quite fast. Flips. It tried to move the boundary 65,536 times per step. So that means that it is run 11,840 times 65,536 individual update attempts, which is quite a few. But it took what? A minute, 2 minutes to run. So even though you're doing a lot of stuff, those things run very fast. Remember that I said it tries to move the boundary, but doesn't always succeed. This number here, and flips again is a bad term, is how many times the boundary actually moved. So out of 65,000 attempts to move the boundary, 1,000 of them succeeded. So about 1 in 65 attempts to move a boundary actually succeeded. So most of the time it tries to move the boundary and decides not to do it. The cost of that numerical stability is a lot of empty calculation, but it still runs pretty fast. Okay. This energy term represents the sum of all of those configuration constraints that we were talking about. The actual numerical value doesn't really matter. Normally, you'll see that number go down a bit, but it doesn't necessarily go down because you can go uphill as well as down. The number of cells here, 164, and inventory, we haven't created any extra cells. But if cells died and disappeared or we created new ones, then the number of cells and the inventory number would be different. Okay. So has everybody got that? One person still doesn't have it working? Everybody got it. Okay. All right. So that is the very basic simulation. Let's come back to Twit and look at our code. Again, we had the size of the lattice, the list of cell types, and then if we look at our volume, for the volume, each cell type can have a different target volume, different compressibility. So we have volume energy parameters for the mesenchymal cell type. Its inverse compressibility is 2. What does that mean? 2. We don't really know. And its target is 50 lattice sites. The surface, again, associated with mesenchymal cell type. Default membrane elasticity of 2. Target surface of 50. And we created an initial blob of cells, radius 51, and the cell width draws squares. They were 49 voxels each, so the initial volume was a little comparable to the actual target volume. And that's the entire simulation. This code creates the simulation that we just ran. And conceptually, what do we have? We have this thing about parallelization, which we can ignore. We have something that defines how big the lattice that we're running on is. We have a list of what kinds of cells we have. And then we define their properties. They have a volume, target volume. They have a target surface area. This says we're going to keep track of their center of mass. We could turn that off if we wanted to. And then we create our initial condition, which was a little circle of cells. That's how the model is defined. I hope that doesn't look too complicated. Things will get more complicated as we do more things. But conceptually, I think the structure of the model, I hope the structure of the model is relatively straightforward. Are there any questions about that so far? I can't tell since I can't see you in the room and people don't have their, I don't force people to turn their videos on. Since I can't see people's faces, it's hard for me to tell whether I'm going unbelievably slowly or too fast or about right. And so if you don't give me feedback in the chat or otherwise, all I can do is use the poll to make sure that people are keeping up. And you certainly can come to me, email me and say, that was too fast, that was too slow if you don't want to speak up. And again, with a diverse class, sometimes it may be there's got to be some compromise. It may be slightly too fast for people, some people, and slightly too slow for others. In the first couple of lectures, I'd rather go slowly and make sure everybody keeps up. Because I don't want people to feel that they're lost. All right, so we've already walked through the various elements and how we can talk about how they correspond to the mathematics. The only thing here that you might want to know about is this draws a circle, position of the circle, center of the circle in the lattice, the radius of the circle in voxels, gap. Sometimes I might want to have my cells initially separated from each other, to have them distributed in space, not adjacent to each other. Gap sets a spacing between the cells. And in principle, if I had more than one cell type, I can create a sort of a mixture of two cell types or more. The one thing you have to watch out for is if I had more than one cell type, I could type cell type 2 here. This won't work because I don't have cell type 2. But one thing that's strange with XML is that spaces have meaning. And so if I create a list of cell types, I have to have no space. I can't have a space between the list of cell types. That's an easy one to have problem. Okay. We've learned how to use player a little bit. Let's go back to player and just review a few things about it. In this case, it's a two-dimensional simulation. So this number here is the slices in the vertical direction. That's not meaningful. I could visualize this as 3D because everything is in fact 3D. Doesn't look very different, but I can rotate it and I see this thin layer that's actually a thin slice in 3D. I'll go back to 2D visualization. The default colors, I don't know what you get, can be pretty ugly. This tool button here lets me change the colors of the cell if I want my boundaries not to be yellow, but to be some other color, like blue, change the color. If I want my cell not to be red, make it yellow. I can change the color. I can play games like that. I can zoom in using the roller on the mouse. Zoom out. If I get lost and I hit R, that will put things back to their default. Okay. I can also do some things like I can save movies. So if I go to this output directory here, I can tell it how often to update the screen. The screen updates actually take some time. So if I'm running very large simulation, I might want to only update every 100 Monte Carlo steps. But I can save images. I can save the raw data sets every so often. The whole variety of saving options. I won't go into all the details right at the moment. All right. Colors we talked about. One thing you might want to do in 2D, you don't do this very often. In 3D, you might want to make one cell type transparent so you can see inside of something. So if you go to the 3D settings, you're allowed to make cell types invisible so that you can see through things. One of the things that happens is that when you stop a simulation, it saves the configuration of the player. This is more of an issue in the desktop than it is when you're running it on NanoHub. But that configuration setting may or may not be what you want when you run a different simulation. And so occasionally you'll have problems, which is the way that the simulation runs on your desktop, well, it may depend on what the last simulation you ran was. And so that's something that one may run into. And if occasionally you'll get an illegal configuration for player and things will crash, and the manuals on CompuCell site explain how to do a reset of that. Okay. If you make a change in Twit here and hit save, and then I go back to player and I hit stop and run again, it should load the latest version. It should update. It's not 100% reliable, and I can't tell you exactly why. So if you made a big change and you don't see it reflected in player, close player and start over. But in principle, player should always load the latest version of the simulation when it runs. Okay. So I think that's it for today. let's just try one thing. There's something called steering, which is something which we could do, which is suppose we go to the volume plugin. Can everybody go to this? And you see the parameters that we typed in? Does everybody go there? If we can change the simulation on the fly. We can change simulation parameters on the fly. We'll find out that not every simulation parameter could be changed on the fly, but some of them can. So suppose we said here the target volume was 50 voxels. What's going to happen, somebody, if I make that target volume 70 voxels, what's going to happen? Somebody have a guess? Not Furkhan, who's done it before, somebody else. Callie, what's going to happen?
If we change that target volume.
Yeah, if I make it 70 instead of 50, what's going to happen?
Let's see.
We could try it
We
out.
may see just an increase in the volume of each cell.
Yeah, but of course, where they're touching each other, they'll be pushing at each other. It's like having all these cells or balloons. I'm blowing into all of them simultaneously, so they're going to have to rearrange. So why don't you try it? Right? Yeah, that's what happened. So that's this idea of target volume versus actual volume. The guys in the middle here are pushed together, so they may be compressed a bit. The ones that are out here on the edge can expand or contract freely.
So the volume does compress. It's not like the cells just take up significantly more space to suit that every cell has that target volume.
Well, you could see the total volume of the tissue did go up. And so the tissue will try to rearrange to satisfy the volume constraint. But over short amounts of time, those cells that are in the very middle are going to be pushing on their neighbors, but their neighbors are going to take time to move out of the way.
Right, okay.
It's the same thing. Suppose that I have a huge crowd of people that are packed into a subway car. I open the door of the subway, they start pouring out. They take up more room and the pressure goes down, but it takes some amount of time to relax. It happens that in this kind of model, that time is given by the speed of sound, but that's not that natural. That may not be a very meaningful thing to do. If I make lambda volume bigger, I'm basically reducing the compressibility of the cells. And if I do that, make that change, maybe a little hard to see, but the fluctuations of the cells will get a little bit less when I do that. And so now you could play with that. You actually have a simulation that is usable. It's not doing that much of interest, but you've got a basic simulation of cells that move around, crawl, occupy space. And you can at least play around with what. Remember, I said if the surface is small compared to the volume, let's make the target surface bigger. See, the cell surfaces get wrinkly because now I have a lot of membrane for my volume. It's like my underinflated beach ball. If I make my target surface very small compared to my volume, they get very stiff. And they stop moving. So I'd like people to play with this simulation. We didn't get that far, perhaps. We didn't do cell sorting, which was where I was targeting. But I hope that gives you something to play with. You can now explore, see everything's frozen, everything's gotten, and you see how things have gotten square. That's that lattice effect that I was talking about. Every cell now is basically behaving like a basketball, and the surfaces aren't able to fluctuate, so I can't rearrange to relax the system. And so one of the things that's nice in CompuCell is that you can do this kind of change on the fly. I don't have to edit the code to change those parameters. See what happens. Okay. Any questions about where we are and what we'll try to Josh and I tomorrow will try, since now that we know how far we got, we'll try to come up with a little homework problem based on this. The main homework problem is going to continue to be thinking about projects and have some ideas for next week. And I hope that that gives you something to play with. Feel free to read ahead and learn how to do things with CompuCell, but at least this is, I hope, a beginning to using CompuCell. One of the things that you'll see also, I guess, before we break, is you see some of the cells are stuck to the wall at the edge. That's one reason to use periodic boundary conditions, and the cells would crawl around the edge and come back on the other side. Because the wall here actually has material properties, so the cells stick to it. Any other questions, comments before we go? No? Well, thank you everybody for sticking around. Thank you for coming back. I'll look forward to, if you want to meet one-on-one, please let me know. If you're having trouble installing CompuCell on your computer locally and you want to run locally, NanoHub was pretty well behaved today, so this was all running on NanoHub. So I hope it worked out for people. But if you're having issues installing CompuCell on your local machine, you want to have it there, please contact Josh and he'll help you with that. Okay. Otherwise, thank you. Good night. Look forward to talking to you next week.